Hello, everybody. For those who haven't met me, my name's Andy. I'm the research lead here in the team. I'm going to be emceeing this event, which are the talks as part of CNS's 20th anniversary. I hope everybody enjoyed the food. Everybody looks pretty happy. Yep, good. Right. Yeah, thank you, Nicole, for organizing. Yeah, so we're going to get started. We're slightly behind, so we're going to get started now. And we're going to start with, so we have a series of brief talks from a variety of CNS and external folks. And we're going to get started with Katy.
Yeah, and these are brief efficiency talks, right? So there are seats here. Please get everybody to come in. And I just wanted to point out that we actually have been moving from building to building to building. And the buildings have different affordances. Like in Luddy Hall, we had a cave system, 3D virtual reality environment. We didn't have this in Bell's, but we had a ton of books in Bell's library. And then here now we have engineering, which is wonderful. And it's also 27 years in Bloomington, which is a beautiful community with lots of farmers markets and lakes and everything else. There is a video embedded here, but we won't have time to run this now, but you can run it later. We will make those slides available later. And you see Lisel in here and many others. It's a cool movie about what CNS goals and ambitions exist. And they are still true today. And Andy did a great job creating that video. All right, 20 years at CNS means a lot of cool teams, a lot of cool people, a lot of cool collaborations, a lot of cool friends, and in some cases family members, and also kids grew up, right? 20 years, that's basically a baby to somebody who has a driver's license and can almost drink. I mean, that all happened also. And so this is kind of cool to see in the photos that you just saw flying by. The kids grow up while we are doing all of this. So it's important to keep that in mind. And some of you might be familiar with those project names. Others might not know any of these. But those were really important abbreviations at the time that was active. And we also have a lot of members. Some of you are back here. It's really wonderful. I think I would not have been able to deal with 300 people back, but I'm very happy to have you all back and here. Michael Ginter today compiled this overview of co-authors from 2006 to 2025. So it's a lot of people doing a lot of cool papers. So thank you for that, Michael. And yes, we cranked out a lot of papers, lots of grant proposals, and ultimately a lot of software artifacts, as you will see. And we had a lot of fun at different projects and went to all kinds of places, small and large. You can try to find me and Philip in here. This was the very first time we met, and that actually created the idea to bring a matria here to Luddy Hall. So you will be able to find both of us in here. Great meeting. 20 years of fun projects, including adventures in Knowledge Land, which you see featured here. And Geoffrey Hobart is right here with us, and he might do future comic books with us. I would love that. And of course, 20 years of publications, lots of papers. Paper. That's what you do if you're in an academic setting. It's also 20 years of teaching. Scott Weingart is somewhere here, right there. He was the man who did the first MOOC together with me in IU's Canvas environment. Very first one at all of IU. And at some point, I had two deans, one on the left-hand side and one on the other end with normal phones. And they both were trying to talk us into it. And we were like, okay, we can do that. And we did that. And then, of course, it's 20 years of tool development. And so that's where Bruce comes in.
Thank you. So my name's Bruce Herr. I'm the technical director of CNS. I started my career with Katy. So I graduated in 2004 as my first job out of college. I worked with Katy from 2004 to 2008 and learned a lot of data analysis and visualization work. And we took that, did a company for till 2017, came back, and then there was a new era. And I was going to talk about the 20 years of development at CNS. So I'm a programmer by heart. So, CNS has always been about data analysis and visualization, but we've also been very passionate about our software to make it usable and extensible and so programmers can use it, but also so it can be used. And so you see that at the beginning I'm going to do an eras tour, and so it's carried on throughout throughout the last 20 years. So I was 24 when I started, and 44 now. So some statistics. We have over 382 repositories that we've had over the last 20 years, probably even more, but that's just what I could find in the different repositories that we have. So we've cranked out a lot of code in the last 20 years and some really great projects. This is just the IV lab. So information visualization lab is what Katy started at CNS or at IU, and then it became CNS. And so I'll talk about the Seychelles era. That's when I came in. Me and Shashi Punamarthi, we devised this great InfoVis cyberinfrastructure software architecture. They used Java and the design patterns at the time. And so we created this. So the idea was that there's all these different algorithms that people use for visualization and analysis. And we brought it together in this tool, the InfoVis cyberinfrastructure. But then we wanted it to grow even further, and we wanted to connect. So our developers who could grab all this data together, and then Seychelles would sit in the middle. This is how it evolved, so that the users could use all these cool algorithms without having to program. So early sort of situation, 2004, but menu driven, but allowed people to do some really cool data analysis and visualization. And so Seychelles was the core that then could be filled up in different ways. So there was Network Workbench, the Herr II tool, and you'll see that these are all these algorithms. So it's like, I forget how many we had at the end, but it was tens, maybe even 100 or more different algorithms that were brought together in this tool. And so the Seychelles was like the empty shell. You could fill with whatever algorithms you wanted. And then Network Workbench had a specific filling, and then Herr II had a specific filling. And this EPIC was epidemics cyberinfrastructure. So each of them were catering to a different community, and that's what made that pretty powerful. And then the HRA era, that's where we're at now, 2017. Been working on that since the beginning, and we've made some really good software. You've probably heard a lot about all these, but we have some really great developers. Students have come in and really made a big difference, and we found ways where they can contribute and then go off to their big careers. But we have really appreciated everyone who's been involved with the software over the years. And I hope that we can continue to make really good software that people can use. All right, thanks.
Colleagues, I'm Philip Beesley, and I want to say thank you so very much for allowing me to be part of your community. It's no exaggeration to say that the dialogues with Katy and the conception of what a macroscope can be have been so fundamental that they've helped create the Living Architecture Systems Group, which is a group of about 160 different people and 60 different institutions. Not a big institution, but many different contributors. I'm going to give a talk which speaks about some changing terms, a couple of different tool sets, 3 quick projects, and I hope that'll be a contribution all in 5 minutes. The oscillation between making something closed and something open translates into the term metastable rather than something stable. And metastable is something we know in our bodies. The sense that instead of asserting ourselves and making something as clear as possible and static as possible, that there's a whole possibility space of gently flexing and moving, which is far from being fragile or diseased or uncertain. Rather, it's a living quality. And yet the terms that we use for boundary and space and potency and power are so very often limited by claims, by domains, by a sense that we need to somehow freeze and render something secure by rendering other things insecure. That kind of disposition for a macroscope, that is a perfect optimal set, is something that I think could be really improved and enhanced by using some very different terms. For example, a very old word for space, ether. Einstein used the word ether. A very different term for the sense of boundaries and claims, our own skins, perhaps care. A very different term for power, potential. Where could something lead? These are terms that are very familiar to biologists and psychologists, But for those that make the built environment and that manipulate information, it seems that our language could shift into a whole different set of terms that are sometimes misunderstood as being weak or even irresponsible or corrupt. The sense that something precarious can be delicious somehow and full of potential and curious and vertiginous is something that I hope to contribute to the world of macroscopes, really drawing from CNS. This work starts then from some practical, deeply physical craft, such as maximizing the potential of physical elements. You can see this kind of form in Amatria, where we stretch and allow things to be intensely strong, tuning things so that they can be as precarious as possible, right at the limit of their ability to stand. And using computational systems, we combine those kinds of elements into basket works, into very, very strong and durable scaffolds, and then combine those again with some pretty interesting interfaces and tools which allow a population of possibility and pluripotency to emerge in this curious kind of jittering, granular kind of way of making, which is rather different than circumscribing an optimum around something. Rather, it's inhabiting potential. Populating that again with behavior, lively kind of matrices of precarious enlivenment, and subscribing to the milieu itself in weather, in influences, where matrices of elements can differentially be influenced and work and intersect together. This turns into a really lovely kind of possibility space where the macroscope is a very, very active design instrument. Here's just a couple of samples of projects taking on the legacy that we've learned from Amatria, from the extraordinary opportunity of being in this community. This is in Delft. We're looking overhead, and you can see some of the rather intensely worked things, but it's by no means a eutrophic swamp. It is very, very distinct and able to be edited. And we'll just I'll just skip through this. I hope that you can see that the gentleness and the unapologetic fragility is one of the features of this work. That is not the same thing as something that will fall apart or that's irresponsible. It's extremely tough. Tough like a goat is tough, or a weed is tough. And I'll just skip to a couple of other projects. One, which is a project of kits, in which the kind of artworks that you're seeing there, that kind of macroscope, is now built out of individual elements, intensely participatory, like the kind of sessions that we used in Amatria, which then turns into an enabling set of open source models and components in which the things that have expressive power are also ones that we can make and that anyone can share in. This then turns into one more project in which those data sets, the underlying digital models, and the rather complex physical environments are twinned together and coupled in order to make living shadows in which data projectors make real shadows, and at the same time, they make pareidolia shadows. That is to say, I'm hallucinating the possibility, I'm dreaming of myself. And the tuning of a real space with a digital space then turns into something which has a very, very curious quality of dreaming data and seeing possibility. So I hope by showing these projects and sharing the kind of legacy of the CNS, I can express my deep, deep gratitude for the term macroscope that Katy introduced to me at Keck Futures on the West Coast many years ago now, and that this might be a contribution that justifies some optimism. Thank you.
Thank you so much, Philip. All right, that's you.
I'm actually going to start passing these around. They're kind of first come, first served, and I may not have brought enough, but if you want to share with your neighbor, that would be great. And you can, and anyone who wants them can keep them afterwards. Hi there, my name is Geoffrey Hobart, and I worked with Katy back in 2009, I think, about 16 years ago. And currently I am working for the American Philatelic Research Library, but I'm also a freelance artist on the side. And let me just click. There we go. So here we are. So we work together on the Adventures in Knowledge Land. I will say we, because Katy is as integral to this project as I was, even though I drew it. And Teresa Theobald also helped with the cleanup art at the end to really get us over the finish line. But the goal of the project was to actually bring the fundamental concepts of information mapping and information systems to children. And I thought that was a really exciting project because it just kind of similar to what I'm seeing today with Philip's work, the emotional response I had when I saw those information maps was very similar. I was just exciting to see vast amounts of data brought into an accessible and a digestible and an understandable form. So I was trying to do that with this comic book. And I hope I did. So a lot of the characters are symbolic of different things that we were talking about. I'll try not to get too deep into it because I only have 5 minutes. But there is a venture capitalist. So that is sort of the, any good science project needs money. There is a scientist and both medical and science are sort of wrapped up in a character called Dr. Scientist. And then there is Curious Gina, which is actually based on one of Katy's daughters. And that's sort of the future of science people. And so we pulled in a lot of interesting natural forms combined with machine forms, and also sort of ultimately the comic book ends with concentrating data into a seed and growing it into a tree of knowledge. And that's sort of the big, big finale, big reveal there. So a lot of sort of coinciding with philosophy, science, academics, and just trying to sort of push all of that, synthesize all of that, and create something that is easily accessible to almost anyone. And I hope we did that. So take a look around, enjoy. If anyone wants to, and everyone can take a copy with that with you. Of course, try not to fight over them because there probably wasn't enough. And I guess I'll pass it on to the next contributor. Thank you so much, everyone.
I guess that would be me.
Laura, are you ready?
Yeah, can you hear me?
Yes, we can hear you.
Okay, can you please advance the slide?
Yep.
Okay, so our motivations for this particular project were to make accessing and retrieving and using public data from open government APIs easier, because I think, as all of us know at this point, it's not exactly as straightforward as the documentation would like everyone to, or the people who wrote the documentation would like you to believe. Would you please advance the slide? So our research question in here was, how can we make public goods data more accessible to public librarians, researchers, and the public? Please advance the slide. So to engage in this process, we're looking at census.gov data. So we engaged in what I guess you could call an old-fashioned domain analysis, which is from Knowledge Organization. So we looked at the census website and some of the query structures, as well as the structure of the API before we started using AI to map this out. So we determined the scope and what we really wanted to look at. We decided we were going to use the American Community Survey for 2023 because that's one of the ones that librarians use a lot. We used retrieval augmented generation because that's been shown to reduce hallucinations, which means we fed the system the data we wanted it to analyze. And then we used the AI and we gave it instructions, and part of RAG is giving it an identity. So we said you are an skilled economist and Python programmer, and you have used the census before. Help us analyze the structure of these things. And what we wanted to extract were the mappings of the tables, the variables, and the concepts. So we used this to then restructure the data, and we went back and verified the mappings. So we then asked the system to generate test queries, and we checked these test queries in the Census API and adjusted as needed. So you need to keep in mind that the Census API is a REST API. So it is you generally type it in the search bar of your browser unless you are programmatically putting it into Python or whatever programming language you use. So it's not sophisticated. And it's really easy to accidentally type the wrong thing in. So we're trying to make this so it's as error-proof as possible. Then we went back and refined this, and we visualized some of the data. And we wanted to test some more complex queries, which required us to query multiple tables and then combine data, as you would see. And we iterated that as necessary. Would you please advance the slide? So part of the problem with the census data in particular is that this goes back to, I guess, before the US was the US, since the time they needed to count people in Virginia, in the colonies. And this was done using paper tables. So the census didn't really go online until 1990. The first version that was available for computers was made available in 1960 for purchase for the price of $10,000. So it was 1990 that you could first get to it at all. And this was as a PDF, which at the time was pretty good. And I know everybody should be groaning internally at the idea of getting the data in the PDF, but that was better than nothing. Please advance the slide. So with these in mind, some of the proposed mappings we got for our question, which was the question we have mapped out here is how many children live with their grandparents in Philadelphia, Pennsylvania. We got some not great answers for table IDs. B09018 was not correct. The one that we needed that we really didn't get was B11003, which was for relationships for married families. There's a more specific name for that. But the AI kind of tried to summarize this, and if you'll advance the slide, you can see that we then were able to visualize, and I used Matplotlib and then later Excel because I had to combine the data and to answer the question. So there's the successful query at the top for grandparents living with grandchildren, but it doesn't include just grandparents who are responsible for grandchildren. That's answered in the table in the bottom and the little orange bit. So that's 4,500 children live with their grandparents, and their grandparents are responsible for them approximately. So married couple families, there are 184,109. Gee, I thought that made that big enough. But we did have to go through and combine some data from different tables. So this is an improvement over what we could ask before from the census and then visualize, but it's still a work in progress. So there's a lot of work to be done here, but I think we can use AI to make this a better process. And this is also a way for researchers and public to better, and librarians to better understand their communities and what their communities may need, especially in terms of library programming, which is what we were getting at with this project. And I think that's my 5 minutes. Thank you.
Hello, everyone. I thought I would talk today a little bit about the Places and Spaces exhibit. It's the Places and Spaces exhibit was first exhibited in April of 2005. So this is also in some ways the 20th anniversary of the Places and Spaces exhibit. So we started with maps of science. I shouldn't say we, I didn't come to this project until much later, but Katy and her cohorts, probably some of you in this room, started with maps of science that could be hung on the wall that represented science in a number of different ways. Let's see, we have been lots of different places since then. Just to summarize, the exhibit has traveled to over 480 venues in 30 countries on 6 continents. We have not yet been to Antarctica, so if you know anybody going who wants to put one in their suitcase, we are happy to send it with you. It showcases the work of 327 maps and macroscope makers. And over the last 20 years, it's been on stages large and small, ranging from the Cannes Film Festival, where the Human Excess short film received awards, to IU Cinema, and to places where big decisions are made, like the Davos Economic Forum. We've been all over the place. Esteemed colleagues from different sectors of information visualization have all joined us annually to review submissions, help us select the best choices for the exhibit, and to put their heads together and reflect with us on the big trends in information visualization as part of our advisory board and our core of ambassadors. Whoops. Let's see. As we continued adding writing object labels for maps that we were hanging on the wall, we kept finding ourselves needing to say things like, and for the interactive version, go to www. Whatever. So a lot of what was happening in information visualization was moving online and becoming digital first in terms of content. And so 10 years in, the exhibit shifted and moved towards showcasing macroscopes, which are interactive visualizations that let us look at things that are too complex or too large, too hard to see with the naked eye. And so these software tools help us slice and dice them and make sense of them, make sense of data by using these interactive visualizations. Let's see. The result is this collection of 40 different macroscopes. If you haven't had a chance to, you're welcome to play with it. It's right outside the door. Let's see. They range in subject matter from catalogs of the cosmos to political discourse, from city smellscapes to the shape of a bird's egg. So it's a very wide range in terms of topic, which we love. And they tackle societal issues such as refugee flows and income inequality, and opioid addiction. So they take on big topics, small topics, a little bit of everything. These new formats were wonderful because people were encouraged to filter data, to zoom in and out, and to really personalize the experience so that they could ask the questions that they wanted to ask and pull out the insights that they were interested in the data. It's been our privilege to share these macroscopes in many different ways. Todd has developed a whole series of interviews with a lot of the makers, which you can find online, and he continues to produce more. So they keep coming out. So that's been a great privilege to speak with the folks who have made these macroscopes. And we've shared a lot of them at in person at science events, at art festivals. They've been on display at the David J. Sensor CDC Museum in Atlanta. Basically, we've taken the exhibit on the road to where the places where people learn and the places where people are exploring. And it's really kind of served as a kernel for people to come together from different disciplines. A lot of people who do information visualization are working in different departments all over a campus. And so this can be a kernel that brings people together so they can learn from each other, which we really appreciate. Let's see. Along the way, we hope that we've moved people, and we hope that we have that we've helped them see the beauty in data. And we hope that we have we've aspired to motivate people to use information visualizations as ways to or tools to improve their lives. But we're not done yet. Oh gosh, there's so many pictures. It was so hard to pick just a few pictures for this. We're not done yet. We're headed into another phase. We've wrapped up macroscopes, and as we transition to the third phase of the exhibit, we look forward to exploring visualizations of different types of intelligence, with an emphasis on cooperation between biological and technological life forms. So to break that down a little bit more, that could include linguistic, kinesthetic, communication, musical, emotional, and all types of intelligences by both biological and technological life forms, with a focus on collaboration and coordination across life forms and intelligence types, to inspire discussions about existing and future sensors and actuators, memory, reasoning, exploration, communication, and shared goals and desirable futures. So broad ranging. I'm really excited to see what kind of submissions we see in May. If you're interested in submitting and have work that you'd like to suggest, we're open to that. We're taking submissions through May 31st. So we look forward to a new era with the exhibit. With that, I'll turn it over to Andy.
Thank you, Lisel. So hi, my name is Andy. I'm the research lead here. My research goal in life is to build these kinds of visualizations, actually a pretty good transition, to not just have them AI generated, but actually make these visualizations and make them explorable for people and explore what the challenges and pitfalls are. So it's about multi-scale data exploration in VR. So my goal is to take the work that CNS does and extend it into 3D and virtual reality spaces, which is also on my name tag here. So this is one of the applications that we are building. It's the H-ray organ gallery in VR, which allows you to stand next to a 3D representation of the human body and explore it. My RAs have, you might see them here with these virtual reality headsets. And feel free to try it out. It's really best explored actually in VR. And we published this on the Meta Quest store recently just to make it accessible to as many people as possible. The base idea behind this was to bring people into the powers of 10 mindset, where you can traverse the human body on a scale in orders of magnitude of 10, and many of you might be familiar with this movie. And this is the design goal behind this application. I did my PhD at IU, and one of the main skills I learned while at CNS was to organize workshops and bring together people that have expertise that I do not have, and vice versa. And so one workshop that I was fortunate enough to organize was at NIAD. They have a huge bio-visualization space full of virtual reality hardware. It's a paradise. And so I had the fortune to work with these amazing other early stage career investigators like myself. They all donated data or gave us data for the organ gallery, and then we visualized that together. And we had a ton of fun. This was last year in October, and this is one of those things where without being at CNS, I don't think I would have ever had the chance or the skills or the contacts, especially to organize this kind of event. We had a ton of fun. It was excellent. And so in this application, we envision the human body as an inverted skyscraper where you start at the top floor, and then every time you go down a level, you get shrunk by 1 order of magnitude. And it's kind of hard to imagine for us because we don't really think usually in powers of 10 how small you suddenly get, even if you just go down 1 or 2 floors. These are some of our amazing collaborators across different universities in the US that have given us data that we then visualized here in virtual reality. This is one of them. This is Hamali Fadnani, who is at Columbia University. This is a 3D stepped relief map that we built in VR. Again, this is really best explored in the headset itself, but it allows you to walk over a piece of brain tissue with 3-dimensional biomarker readings poking out of the floor. This is another scene that we built with Yang Miao from Duke University. This allows you to explore different cells on different layers, visualized with different colors. You can always see the data visualization roots in here, but now taken out into 3D and virtual reality spaces. So I think this is how this all connects into the work that CNS in general is doing here. This scene is also deployed into the organ gallery on the headset here. And then I just made this visualization this morning. We, of course, also use GitHub, just like Bruce's team, to develop our code. And this is just one code repository. This is an animation for the organ gallery that shows how the code has developed. So you see a thing like this human floating around. So that's me making a bunch of GitHub commits. And then this just visualizes how these commits are coming in. And this goes about 1/10 of a second per day. And so you can see at some point there's like multiple people floating around. That's whenever I have team members that then help develop code with me. And then we all kind of float back and forth and change different things and add new functionality, etc. Sometimes it's rather quiet and it's just me hovering around, adding new functionality here and there. Other times it's like 4 or 5 people that collaborate together with me. And so you can kind of nicely see the flow over time. This is about 4 years old at this point, this repo. And it's just one repository. So if we take all 325 that Bruce mentioned, this is going to get out of hand very quickly. But yeah, it's just very nice to see how, it grows and it shrinks as time passes by. And now we're in July 2024. And you can see I have some people that work with me now, then it's me again, and suddenly it explodes again. So yeah, data visualization. Telling a good story here. Okay, this is a backup thing that I had made. Here we go. Quick challenge for you. in the organ gallery, Easter is coming up. We hid an Easter bunny in there, a 3-dimensional fella. So if you go into VR, try to find it. If you tell me on which floor it is, there might be a little surprise. And then I want to finish with some pictures. So I went back into my personal archives, and these are pictures from my first year at IU. When I first joined CNS, I was working a lot with cameras and films. So we made a bunch of we made an image video about what CNS is and does. We made a video for our information visualization class about map makers. This year we shot at the IMU with David Kloster, who was a grad student, just being dressed up from the opera, I think. It was just a ton of fun. This is from that shoot earlier that Katy was showing in her slide deck in the media school. So we really got around. I think we went to 4 or 5 different spaces on campus where you could do professional video development, and had a ton of fun just shooting a bunch of content. This is also, I think, from my first year at IU. It's actually surprising how many people are still here. So I see Samantha here, and yeah. So it's just nice to see that there is consistency here. This was Social Science Research Commons, I believe. And then this picture, I don't know what we were doing. We're just being silly. So Philip was here. We had been spending 2 days planning something. I think building those kits that you can see in the background there, those dendrite kits. And we were just shot. It was like a Sunday morning. We're just really tired, and then we got silly, I guess. So yeah, there were always fun times here at CNS. Okay, thank you. That's me. Yeah, talk to me. All right, Bruce, coming to us from Cincinnati.
Thanks, Cindy. It's such a treat to be here. I'm just I love being an adopted family member. You guys are just an exquisite place, and the philosophy behind how you're approaching it, trying to work with artists and poets and engineers, and it's very enriching, and the opportunity is just it's wonderful. So when I talk to you, I'm in Cincinnati Children's. I've been there for 40 years, and it's just like going to Disneyland every day, sort of like coming here. And the thing that is so motivating for me is talking to a mom who's got a kid that is really sick and can't figure it out, or the clinicians that are trying to get their data together in a cohesive way, and they really trying to understand the system. How do you formulate an approach to understanding the complexity of disease, a non-functioning system, a dysfunctionality, and so there are paths, and so my goal is to try and do knowledge-based reasoning across multimodal data, and to bring bottom lines to our understanding of systems, and make all these aspects of data, all aspects of knowledge. I love the library science presentation, and all of that can help you reason and understand more, and we can program agents to do what we want to do, and do it even better and more carefully than we do. But I want to show you a manual approach to that, and trying to understand a biological system, and just how do you get different information types to work together. So think about, from a gene point of view, we have all these different knowledge areas about genes. There's gene ontology that talks about what's the molecular function class. Is it an enzyme? Is it a structural protein? Is it where is it localized? Is it cell in the cell surface, or in the nucleus, or associated with inner or outer membranes, or just out in the matrix? Oops, sorry. Anyway, so you can track through all these different information types, and you can start anywhere if you want to. And everything that is a discrete entity that's described as a biological process, cell cycle, or lung development, or breathing, all of that, the cardiovascular function in response to exercise. All of those things are a process, and you can dissect that, and there's annotations of genes and pathways that relate to that. You can go into PubMed, grab that, and start to build a model that includes genes and cell types and tissues that have to work together, signal to each other, and then you can build models of systems at any level. And so you can start anywhere and learn incredible things. So let's talk about like cell atlas stuff and molecular profiles and transcriptome. You get genome-wide information about gene expression in different cell types. There's a lot of different things, epigenomics, all these different kind of ways of linking sets of genes under conditions and cell types and cell types that are working with each other to accomplish a task. And it could be to form the structure, to support the functions of the structure, to withstand, like the slings and arrows of outrageous infectious diseases or trauma. And then, that idea, I was actually thinking in the Amatria, that the robustness of the system that you develop. So that's something that's evolved, over many billions of years, is the robustness of biological systems, that there are these compensatory mechanisms. We can find those, but when you have mutations that really cause a problem in a system, just a single base change of a hemoglobin molecule that causes it to sickle, there's so many downstream consequences of that one molecular change. So you can build network in there, and you can extrapolate to fetal hemoglobin suppresses the polymerization of that. So now I need to understand epigenomic regulation of fetal hemoglobins, and one of the key treatments for sickle cell hemoglobin is to prevent this inactivation of the fetal gene. So you can reason across all these pathways, and deal with complex and you can put them together. So let's take the cell atlas information. And I just want to go one step deep into cell atlas. And this is a view. This is sort of the ASCT plus B beyond. So you can't, the ASCT plus B gets you markings of these individual cell types, but they don't really, if you want to understand the biology of one of these cell types that occurs within a category of a cell type, this happens to be excitatory and inhibitory neurons. But there are these different levels of hierarchy of designation. These are all of the neurons. And then these are the excitatory and these are the inhibitory neurons. And then these are the subclasses of them. So that when these are 200 genes that are kind of summarizing some level of determination of the state at these different levels of cellular differentiative hierarchy. And so this set of genes right here, they don't distinguish between excitatory and inhibitory neurons. If you just jump to one of these, SST positive neurons, you're going to completely lose track of the fact that these genes are not going to work in that cell type unless those genes are present there for them to interact with in a network. So you've got to build these networks in a hierarchical way. Otherwise, you're going to, you can't. The jobs of these is not to determine the full state of that. The jobs of these are to modify the biology of these modules. And you can do that, again, hierarchically at many levels. And so you can build, mechanistic models. So you go, the key thing is to go from this kind of cloud view of expression pattern or some, epigenomic information or something about, modification state of proteins to the modularized view. And then you can work in networks about really trying to understand these entities and how they work with each other. So I'll just jump ahead to one. This is just one cell type in cell. So I'm a PI of the Data Coordination Center for the Human Lung Map NHLBI project. And this is these three cell types over there are they're called aerocytes. They're capillary endothelial cells that have specifically differentiated to in the alveolus to allow for air to come from your alveoli and across your blood. So it's a very specialized endothelial cell. And then we can see three different subclusters of them. And this is just like 180 genes. But without these 180 genes working together, you wouldn't be breathing very happily. And there's so much that's done. And I'm sorry, I didn't put it in the higher level, above this would be the hierarchy of other endothelial, pan-endothelial genes or capillary endothelial genes. But this is a pretty good representation of what has to happen from those cells in three classes. And so what you do is go into an enrichment analysis, and it's like just a knowledge graph based approach for all those different categories that I was showing you around, genomic function, gene function and gene networks. And then you start to see, oh, there's tremendous enrichments for vascular, depending on the category, in GO and HP and MP, loss of blood vessel formation and structuring and the biological process. If you put all of those together, okay, and those are genetic associations. And then these are functional associations, like tons of genes involved in cell surface, anchorage. How do these cells work? What do they have to accomplish in order to work in situ to let you breathe and let you breathe happily? It's a wonderful thing. It's a very wonderful thing. And when it doesn't happen, that's, misery. And so you want to be able to there's problems that happen right after birth. There's, if you're born early, if you have a genetic condition, and then you progressive loss of breathing, these are miserable conditions. So we need to really understand how to protect those cells and build them up, and lots of things, tasks on that. So these are all the molecular functions that link them. So we're trying to then get a network view of what is it that allows these cells to accomplish it. And it's all this biology. So you have these subspaces of functionality that are there to, account for the really specialized tasks that these cells have to do. They've got to be very thin, very, very thin to allow gas exchange across, and they've got to adhere very tightly to the alveolar epithelial cells that, bring in the gases. And so putting all of that information together, of that 179 signature, only 106, and this is pretty good. This is a lot of prior knowledge, but you say anything having to do with any endothelial, and maybe it doesn't, it wasn't specialized information about that erythrocyte endothelial cell, but it's any known association with endothelial cell biology, including all PubMed references and all GO and all mutations, only accounts for 106 out of that 179 signatures, leaving these 71 genes unaccounted for. But to think that these guys, are they just epigenes? No, this is discovery space. And so we link that, the priors with the observations, and then we can really build up, it's a very easy workflow to go, okay, what are the most important discoveries? Which of these, this is, massively important regulatory genes. This is, just a lot of these things. So how do they fit in the networks that are, deterministic of the functionality there? So just stepping a couple things through it, and it's a big network projection, and you want to reason with it, and then you want to see, okay, what's the problem in the context? And then, you have to specialize on the different tasks, the apical, the basal, the exposure to oxygen. That's a very high thing. Anyway, so here's how, just within a set of genes, how a very important task for those cells is now also part of the, new genes fit into that. And so you fit these genes into the existing networks. And so then they give you an extra contextualization of those networks by them. And it's like taking candy from a baby. It's so simple how these discoveries work. But biological investigators don't get it. And so it's up to us, as data scientists and out of the box thinkers to give these paths to discovery to the users. So that's, you guys are, stewards of data and knowledge, and, being, a cultivation space, it just reminds you of driving out here in the beautiful nature that it's, you guys, it's a special place, and you have a special ability. So anyway, so one aspect that comes out in this kind of systems view is you can now see on large-scale drug screening databases how some of the key signatures of that cell to become that cell are actually very strongly enriched by, in this case, it's top 6 drugs that can activate some of those signatures. So now this is an immediate, it's a little bit of an out of context thing. It's working in fibroblasts or neural cells, but it's pretty damn good. And these are, and some of these, I guess half of them have actually been in clinical trials at some level to validate. So this, this idea of then, having hypothesis generation that's actionable, targetable, usable for preclinical testing. That's, that's the basic kind of our potential. If we, by putting this data together, putting this knowledge together, really linking it up, and then allowing for a user to, what's my problem? What am I trying to solve? What's my opportunity? And then, where could it apply? All of that stuff, I think, is goes to the impact of what we're doing, and it's great to be here.
Thank you. All right, Nikita, are you ready?
Yeah. Hey, everyone. And it's really great to be here. And so I'm an engineer, artist, and scientist, and I do different data visualization projects, often experimental. And one of my projects became a part of this microscope exhibit, which I'm really grateful for. And just before that, it won a data visualization, World Data Visualization Prize in 2019. So today I just wanted to remember the project and do a quick demo. Since it's an interactive project, I thought I could maybe take over the screen sharing and just walk you through.
Yeah, sounds good.
Yeah, so here we go. The project is called an alternative data-driven country map, which is more like actually a world map. And the idea behind the project is we can describe different countries through different metrics, different indices, like GDP, population, surface area, happiness index, and so on and so forth. So in this project, me and my team, we thought that why won't we take this data and we use an algorithm called TSNE, which we probably heard about. This is a very powerful algorithm that can, if you have a multidimensional data set where every point has many, many, many variables, you can just blindly feed your data set into this algorithm and it will reduce the number of dimensions, and it will cluster similar points together, but it won't tell you like what is the specific criteria for clustering. You kind of like have to go and explore it yourself. So here in this project, you can observe it in real time because the algorithm runs right in the browser. So if I run here, you will see the algorithm calculating the clusters right now, right here, and then it shows you this data-driven world map where you could freely zoom in and explore different countries. And so, as I said, they're clustered together based on a number of different metrics like population, GDP, Happy Planet Index, and many, many more. And different colors represent different continents. So you can see the legend on top. And here we built in this kind of a data journey, a data storytelling experience, where we tell people a little bit about the algorithm, and then we explore the canvas a little bit. So one interesting observation here is that we have this dedicated cluster of countries, and these are all countries which we call developed countries. And it's really an isolated cluster on this data plot. And if you go deeper and try to go through the data and understand why, you will see that these countries are characterized by high values in indicators like political stability, control of corruption, judicial effectiveness, etc. And then you can see that India and China are close together. Well, they close also geographically, they close by population. So then you have a cluster of Middle East countries, but for some reason Singapore here as well, and Brunei. So why? We don't know yet, but you can go and explore. Then we have Russia and Brazil, also unusual neighborhood. But then what we really proud of, we built in this feature where you can actually uncheck the metrics you want to use. And then once I uncheck the population and surface area, Russia and Brazil, they goes apart and they end up in two different clusters. And then we suggest you to play with this metrics. And here we say, like, why don't we try to take, for example, the US and reduce the amount of the number of health expenditure. So you can see the sliders go down, and then right away, US goes from the cluster of developed countries into some other cluster. Now it's close to India and China. So this kind of like a gamification kind of experiment, allowing you to play with these indicators to see how t-SNE works and maybe find some unexpected consequences. Yeah, so if you are interested, this is the URL. I think I have added it to the slides as well. So you can just, after this, you can go and explore and maybe you'll find something unique, something unusual here. That's it. Thanks.
Thank you very much, Nikita. All right, I think we have one final slide that basically says Amatria, and I think that's also the last slide of the event. Thanks, everybody. Thank you to everybody who gave a talk, including the people online. You want to say some last words, Katy?
Totally. I love last words. So yeah, I saw who's online. Thank you for coming. I see you all here in the room. It's wonderful to have you all here. And I think there will be new collaborations, new friendships, new exciting innovations coming out of this one day. Thank you for coming all. It was wonderful to see you here. There's more food and a beautiful, beautiful flower basket over there. We will share photos later on. And just enjoy. And come back in another 10 years, maybe. Not 20 years, maybe. All right. Thank you. Bye.